Cell, the unit of life. NCERT based single sentence questions. Let's start. The theory which has created a sense of mystery around living phenomena, that is physiological and behavioral process, is cell theory. Organisms composed with single cell only are called unicellular organisms. Unicellular organism. This mystery was the requirement of integrity of cellular organization for living phenomena to be demonstrated or observed. Cell theory. Organisms which are composed with many cells are called multicellular organisms. In studying and understanding physiological and behavioral processes, one can take a physico-chemical approach and use dash to investigate. Use cell-free systems to investigate. If a single cell perform all the essential functions of life and have an independent existence, such organisms are called unicellular organisms. Unicellular organisms. Fundamental structural and functional unit of all living organisms is cell. This approach enables us to describe the various processes in molecular terms. Physico-chemical approach. Physico-chemical approach. The invention of the microscope and its improvement leading to the electron microscope reveal all the structural details of the cell. All the structural details of the cells. The approach is established by analysis of living tissues for elements and compounds. It will tell us what type of organic compounds are present in living organisms. Name the approach in which the above statement mentioned. Physico-chemical approach. Physico-chemical approach to study and understand living organisms is called reductionist biology. Scientists who first saw and described a live cell, Anthony von Leeuwenhoek. Scientists who discovered the nucleus, Robert Brown. German botanist examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. Matthias Sladen in 1838. A British zoologist studied different types of animal cells and reported that cells had a thin outer layer, which is today known as plasma membrane. Theodore Schwann. Scientists who concluded that based on his studies on plant tissues that the presence of cell wall is a unique character of the plant cells, Matthias Sladen. In 1838. Scientists who proposed the hypothesis that the bodies of the animals and plants are composed of cells and products of cells. That is Theodore Schwann. Scientists who together formulated the cell theory. Theodore Schwann and Matthias Sladen. Scientists who explained that the cells divided and new cells are formed from pre-existing cells. Rudolf Virchow in 1855. Cell theory, which is formulated by Schwann and Sleeden is not able to explain this main feature. This is not explain this main feature. How new cells are formed. They are not able to explain how new cells are formed. Omnis cellula e cellula means. Omnis cellula e cellula means new cells are formed from pre-existing cells. Scientists who proposed the theory omnis cellula e cellula, Rudolf Virchow. Scientists who modified the hypothesis of Sleeden and Schwann to give the cell theory a final stage, Rudolf Virchow. Cell theory, as understood today, is all living organisms are composed of cells and products of cells. Then all cells arise from pre-existing cells. A typical plant cell used in experiments which has distinct cell wall has its outer boundary and just within it has a cell membrane, onion peel. A typical animal cell used in experiment which have an outer membrane as a delimiting structure of the cell. Human cheek cell. Inside each cell is a dense membrane-bound structure called nucleus. Here we have answer. Cytoorganelle which contain the chromosomes and genetic material and DNA. Nucleus. Cells that have membrane-bound nuclei are called eukaryotic cells. Cells that lack a membrane-bound nucleus are called prokaryotic cells. Various chemical reactions occur here to keep the cell in the living systems. Cytoplasm. In cytoplasm. Membrane-bound cytoorganelles are endoplasmic reticulum. Membrane-bound cytoorganelles are endoplasmic reticulum. Golgi complex. Endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles are all having membrane-bound cytoorganelles. Non-membrane-bound cytoorganelles are ribosomes. Non-membrane-bound cytoorganelles are ribosomes. Location of ribosomes in plant cells: cytoplasm, mitochondria. Location of ribosomes in plant cells: cytoplasm, mitochondria, on rough endoplasmic reticulum and chloroplast. Rough endoplasmic reticulum and chloroplast are having ribosomes. Location of ribosomes in animal cells: that cytoplasm, mitochondria, and rough endoplasmic reticulum. Identify the given characteristics to whom they belongs to, prokaryotic or eukaryotic. A semi-fluid matrix called cytoplasm occupies the volume of the cell, both. Contain membrane-bound distinct structure called organelles like endoplasmic reticulum, Golgi complex, lysosome, mitochondria, microbodies, and vacuole. It is eukaryotic cell. Cytoplasm is the main arena of cellular activities in both bacteria, blue-green algae, mycoplasmia, and PPLO, pleuropneumonia-like organisms are prokaryotes. Lack membrane-bound cytoorganelles, prokaryotic cells. Non-membrane-bound organelle ribosomes are present in non-membrane-bound organelle ribosomes are present in both. Smaller and multiply rapidly are Smaller, multiply rapidly are prokaryotic cells. They include the protist, plant, animal, fungi, eukaryotic cells. There is no well-defined nucleus, prokaryotic cell. Ribosomes are associated with the plasma membrane of the cell in prokaryotic cell. Nuclear membrane is present, eukaryotic cell. Reserve food material inclusion bodies, prokaryotic cell. Cells with an extensive compartmentalization of the cytoplasm through the presence of membrane-bound organelle, eukaryotic cell. Their genetic material is organized into chromosomes, eukaryotic cell. Contains 70S ribosomes only, prokaryotic cell. Possess an organized nucleus with a nuclear envelope. Possess an organized nucleus with a nuclear envelope, eukaryotic cell. Most rare cells, particularly the bacterial cells, have a chemically complex cell envelope. Most prokaryotic cells. Cells have a variety of complex locomotory and cytoskeletal structures, eukaryotic cell. Ribosomes are associated with the plasma membrane of the cell, prokaryotic cell. Non-membrane bound organelle called centrosome is present in animal cell. Largest isolated single cell is egg of ostrich. Longest cell of animals, nerve cell. Smallest cell, mycoplasma. Cell organelle helpful in cell division of animal cell, centrosome. Cells differ greatly in size, shape, and activity. True. Cells also vary greatly in their shape. True. Eukaryotes are generally smaller and multiply more rapidly than prokaryotes. False. Prokaryotes are generally smaller and multiply more rapidly than eukaryotic cell. True. Prokaryotes are generally the shape of the cell may vary with the function they perform. The shape of the cell may vary with the function they perform. True. Organisms with 0.3 millimicron in length, mycoplasmas. Organism with 3 to 5 millimicron in length, bacteria. Among multicellular organisms, these cells are about 7.0 millimicron in diameter, red blood cells. Rod-shaped bacteria, bacillus. Spherical-shaped bacteria, coccus. Comma-shaped bacteria, vibrio. Spiral-shaped bacteria, spirillum. The specialized cells of the specialized part of the prokaryotic cell is fundamentally dash even through prokaryotes exhibit a wide variety of shapes and function. Similar. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The fluid matrix filling the cell is called cytoplasm. Condition of nucleus in prokaryote. There is no well-developed nucleus. Well-defined nucleus. There is no well-defined nucleus without nuclear membrane. You can see the chromosomal content in cytoplasm. Condition of nucleus in eukaryote. Specified nuclear membrane is present, which makes them to call as eukaryote. Condition of genetic material in prokaryote. Naked, not enveloped by a nuclear membrane, not enveloped by a nuclear membrane. In addition to the genomic DNA, in addition to the genomic DNA, many bacteria have small dash DNA outside the genomic DNA. Many bacteria have small circular DNA, call it as plasmids. Plasmid is small circular DNA. Small circular DNA. Plasmid is a small circular DNA present outside the genomic DNA. This DNA confers a certain unique phenotypic characters to bacteria, plasmid. Special character which bacteria attain due to some genes on plasmid. Special character which bacteria attain. What is the special character? That is resistance to the antibiotic. Ampicillin and tetracycline resistance against. This DNA used to monitor bacterial transformation with foreign DNA, plasmid. No organelles like the ones in eukaryotes are found in prokaryotic cell except for ribosomes. A specialized differentiated form of cell membrane is called in prokaryotes. A specialized differentiated form of cell membrane is called mesosome. Then thin filamentous extensions arise from the cell wall of motile bacteria are called flagella. Type of ribosomes in prokaryotes. 70S ribosomes are present in prokaryotes. Prokaryotes are having 70S ribosomes. Essentially enfoldings of cell membrane are in prokaryote, that is mesosome. Phosphate granule, synophysian granule and glycogen granules, whom we call inclusion bodies. Prokaryotes have something unique in the form of inclusions. Example, prokaryotes have something in the form of inclusions. Example, mesosome. Three parts of bacterial flagellum are filament, hook, basal body. They are about 15 nm to 20 nm in size and are made up of two subunits called 50S and 30S subunit, which when present together form 80S subunit, that in prokaryotic ribosomes. 50S, 70S, here we have 70S subunit. 50 plus 30, 70S subunit will form. Mesosome is a characteristic feature of prokaryotes. Number of subunits in ribosomes, 2. In some prokaryotes like cyanobacteria, in case of cyanobacteria, membranous extensions into the cytoplasm are called chromatophores, which contain pigments. Number of tightly bound layers in chemically complex cell envelope of prokaryotic cells. 3. Outermost layer of the cell envelope in prokaryotic cell, glycocalyx. Middle layer of the cell envelope in prokaryotic cell, cell wall. Inner layer of cell envelope in prokaryotic cell is plasma membrane. Composition of glycocalyx in different bacteria, same or different. The layer of cell envelope which determines the shape of the cell, that is cell wall. All the three layers of cell envelope act together as a single protective unit. Vesicles, tubules, lamella are the extensions of this layer of cell envelope, plasma membrane. On the basis of differences in the cell envelope and the manner in which they respond to the staining procedure developed by Gram, bacteria are classified as gram positive and gram negative bacteria. The name of scientists, gram positive and gram negative bacteria. Structures which help in cell wall formation, DNA replication and distribution to daughter cell. Extensions of plasma membrane. It could be a loose sheath called slime layer in some, while in others it may be thick and tough called the capsule. It means, what is the meaning of it? That is glycocalyx. Glycocalyx could be a loose sheath called the slime layer in some, while in others it may be the thick and tough called the capsule. This membrane is similar structurally to that of eukaryote, plasma membrane. Layer of cell envelope which provide a strong structural support to prevent the bacterium from bursting or collapsing. Cell wall. A special membranous structure is the dash which is formed by the extensions of the plasma membrane into the cell, that is mesosome. Structures which help in respiration and secretion processes are structures which help in respiration and secretion processes are extensions of the plasma membrane. Extensions of plasma membrane. Bacteria that take up the gram stain are called gram positive bacteria. The cell envelope, which is selectively permeable in nature and interacts with the outside world, plasma membrane. Bacteria that do not take up the gram stain are called gram negative bacteria. Structures which increase the surface of the plasma membrane and enzymatic content. Extensions of plasma membrane. Bacterial cells are motile or non-motile? Maybe motile or non-motile. Membranous extensions of cyanobacteria which contain pigment, chromatophores. Longest portion of bacterial flagellum which extend from the cell surface to the outside, that is filament of flagellum. Surface structures of the bacteria but do not play a role in motility are pili and fimbriae. Elongated tubular structures made of a special protein called pili. Small bristle-like fibers sprouting out the cell are called fimbriae. In prokaryotes, ribosomes are associated with plasma membrane of the cell. Reserve material in prokaryotic cell which are stored in the cytoplasm are called inclusion bodies. These structures are known to help attach the bacteria to rock and streams and also to the host tissues in some bacteria. These structures are known to help attach the bacteria to rocks in streams and also to the host tissues in some bacteria. Fimbriae. These are not bound by any membrane system and lie free in the cytoplasm phosphate granules. These are not bound by any membrane system and lie free in the cytoplasm, that is phosphate granule, synaphysian granule, and glycogen granule. Inclusion bodies. If several ribosomes attach to a single mRNA and form a chain, they are called polyribosome or polysomes. Polyribosomes are polysomes. Gas vacuoles are found in blue-green and purple and green photosynthetic bacteria. Blue-green and purple and green photosynthetic bacteria. Ribosomes of a polysome translate mRNA into protein. The eukaryotes include eukaryote examples: protists, plants, and animals. Protists, plants, animals, and fungi. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm due to presence of membrane-bound cytoorganelles. Nucleus in eukaryotic cells is organized with a nuclear envelope. Eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Genetic material of eukaryotic cell is organized into chromosomes. All eukaryotic cells are identical or non-identical? Non-identical. Presence of cell wall and plastids is the characteristic feature of plant cell or animal cell. Presence of cell wall is a characteristic feature of plant cell. Centrioles are present in animal cell, which are absent in almost all plant cells. Presence of a large central vacuole is the characteristic feature of plant cell. The detailed structure of the membrane was studied only after the advent of the electron microscope in 1950s. Chemical studies in the cell membrane, especially in DAS cells, human red blood cells, enable the scientists to deduce the possible structure of the plasma membrane. Composition of cell membrane according to chemical studies, composed of lipids and proteins. Major lipids that are arranged in bilayer are phospholipids. Major lipids which are arranged in bilayer phospholipids. The lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tails towards the inner part. Hydrophilic portion in lipid bilayer head. Hydrophobic portion in lipid bilayer tail end. Hydrophilic portion of lipids are arranged within the membrane towards outside. Hydrophobic portion of lipids arranged within the membrane towards inside. This ensures that the non-polar tail of saturated hydrocarbon is protected from the aqueous environment. Arrangement of tail end towards the inner side of the membrane. Other components observed in the cell membrane along with phospholipids, that is cholesterol. Biochemical investigation clearly revealed that the cell membrane also possesses these substances along with phospholipids and cholesterol, that is protein and carbohydrate. The ratio of protein and lipid dash considerably in the different cell types, yet considerably they vary. Percentage of protein and lipids in the membrane of erythrocyte. Protein 52%. Protein 52%. Lipids are about 40%. It helps in cell to cell interaction and provide barrier to undesirable macromolecule. Cell wall. Depending on the case of extraction, membrane proteins can be classified as integral proteins and peripheral proteins. The proteins lie on the surface of the membrane are peripheral proteins. If the protein which partially or totally buried in the membrane are integral proteins. An improved model of the structure of the cell membrane was proposed by fluid mosaic model. Fluid mosaic model. An improved model of the structure of the cell membrane is proposed by fluid mosaic model proposed by Singer and Nicholson in 1972. The model is fluid mosaic model. An improved model of the structure of cell membrane, that what we said, that is fluid mosaic model. This ability to move within the membrane is measured in fluidity, quasi-fluid nature. This is also important from the point of view of functions like cell growth, formation of intercellular junction, secretion, endocytosis, cell division. The fluid nature of cell membrane. One of the most important functions of the plasma membrane is the transport of molecules across it. Transport of molecules across it. It gives shape to the cell and protect the cell from the mechanical damage and infection in fungi and plant. Cell wall. Nature of permeability of cell membrane to molecule present on either side of it. Selectively permeable nature. Selectively permeable nature. Non-living rigid structure which forms an outer covering for the plasma membrane of fungi and plants is called, that is, cell wall. In the cell wall of a young plant cell, the primary wall is capable of growth. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes move across the cell membrane along the concentration gradient from higher concentration to the lower. Such process is called simple diffusion. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. The polar molecules cannot pass through the non-polar lipid bilayer. Hence, they require carrier proteins. Of the carrier proteins, they require membrane to facilitate their transport across the membrane. Example for active transport, that we have sodium and potassium pump. A few ions or molecules are transported across the membrane, again is transported across the membrane, again is a few ions or molecules are transported across the membrane, again is their concentration gradient, that is from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. Cellulose, galatins, mannans, and minerals like calcium carbonate is the composition of cell wall in algae. Cellulose, hemicellulose, pectins, and proteins is the composition of cell wall in plants. Cell wall of a young plant cell, the primary wall is capable of growth, which gradually diminishes as the cell matures and the dash and the growth is formed on the inner secondary wall membrane, formed in the inner side of the cell. That means that the secondary wall membrane towards the side of the cell. Secondary wall membrane towards the side of the cell. Middle lamella of cell wall mainly composed of calcium pectate. It holds or glues different neighboring cells together. It holds or glues different neighboring cells together. Calcium pectate of middle lamella. The cell wall and middle lamella may be traversed by dash which connect the cytoplasm of neighboring cell, plasmodesmata, while each of the membranous organelle is distinct in terms of its structure and function. Many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosome, and vacuole. The endomembrane system include endoplasmic reticulum, Golgi complex, and that reticulum, that is the statement is true or false. True. Since the functions of mitochondria, chloroplast, peroxisomes are not coordinated with the above component, these are not considered as a part of the endomembrane system. Okay. Statement is true or false? True. Mitochondria, chloroplast, endoplasmic reticulum, Golgi complex, peroxisome, lysosome, vacuole select the organelles which are the part of endomembrane system. Endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Electron microscopic studies of eukaryotic cells reveals the presence of a network of reticulum of tiny pipe-type tubular structures scattered in the cytoplasm that is called endoplasmic reticulum. Cell organelle in which intracellular space divided into two distinct compartments, that is luminal and extraluminal compartments. The two distinct compartments are seen in endoplasmic reticulum. Space inside the endoplasmic reticulum is called intraluminal space. Space outside endoplasmic reticulum in between the network of cisternae in the cytoplasm is called extraluminal space. Location of attachment of ribosomes on ER, outer surface. Endoplasmic reticulum bearing ribosomes on their surface are called as endoplasmic reticulum bearing ribosomes on the surface is called rough endoplasmic reticulum. Such endoplasmic reticulum is called rough endoplasmic reticulum. In the absence of ribosome, they appear smooth. Endoplasmic reticulum is called, such endoplasmic reticulum is said to be smooth endoplasmic reticulum. Endoplasmic reticulum, which is frequently observed in the cells actively involved in protein synthesis and secretion, rough endoplasmic reticulum. They consist of many flat disc-shaped sacs or cisternae of 0.5 millimicron to 1.0 millimicron in diameter. Golgi complex. Endoplasmic reticulum, which is extensive and continuous with the outer membrane of the nucleus. Rough endoplasmic reticulum. Rough endoplasmic reticulum. Cytoorganelle, which is stacked and parallelly arranged. Cytoorganelle, which is stacked and parallelly arranged. That is Golgi complex. Major sites for the synthesis of lipids is smooth endoplasmic reticulum, densely stained reticular structures near the nucleus, later named as Golgi complex. In animal cells, lipid-like steroidal hormones are synthesized in smooth endoplasmic reticulum. Scientists who first observed densely stained reticular structures near the nucleus, that is Camillo Golgi, identified, and it was now named as Golgi complex. Number of cisternae in the Golgi complex, constant or varied? Constant, they are not constant, they are varied. Important site of formation of glycoproteins and glycolipids is Golgi complex. Protein synthesis is done by ribosomes. Synthesized protein stored in cisternae of Golgi complex. Sorry, see that synthesized proteins are stored in cisternae of rough endoplasmic reticulum. Cisternae of RER release proteins in the form of small packages called vesicles. Vesicles of endoplasmic reticulum fuse and form cisternae of Golgi complex. Cisternae of Golgi complex, which is near to the cisternae of endoplasmic reticulum, are called cis phase or forming phase. Cis phase or forming phase. Cisternae of Golgi complex, which is near to the plasma membrane, transphase or maturing phase. Forming phase of Golgi complex is towards cisternae of forming phase of Golgi complex is toward cisternae of RER. Maturing phase of Golgi complex is towards plasma membrane. Cytoorganelle, which is principally perform the function of packaging materials and to be delivered in the intracellular targets, are secreted outside the cell, that is Golgi complex. Materials to be packaged in the form of vesicles from the ER fuse with the space of the Golgi apparatus and move towards the maturing phase. Condensation, modification, packaging of synthesized protein occur in this cytoorganelle, Golgi complex. Condensed, packed, modified proteins are released in the form of vesicles from Golgi complex are called lysosomes. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. Materials to be packaged in the form of vesicles. Materials to be packaged in the form of vesicles from the ER fuse with the cis phase. ER with the fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. The number of proteins synthesized by ribosomes on the number of proteins synthesized by ribosomes on the endoplasmic reticulum. Endoplasmic reticulum modified in the cisternae of the endoplasmic reticulum before they are released. Its transfer number of proteins synthesized by ribosomes and the endoplasmic reticulum are modified in the cisternae of the Golgi complex before the release from its transphase. The vacuole is bound by a single membrane called tonoplast. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus, that is lysosomes. Cytoorganelle, which contain water, sap, excretory product, and other materials not useful for the cell, that is vacuole. Cytoorganelle, which produce cellular energy in the form of ATP, mitochondria. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes like hydrolases, lipases, and proteases, then carbohydrates that we call amylases. Membrane bound space found in the cytoplasm is vacuole. Enzyme of lysosomes, which are capable of digesting carbohydrate, protein, lipids, and nucleic acid. The hydrolases like lipases and proteases, carbohydrates, amylases. Vacuoles occupy 90% of the volume of the cell in plant cells. It facilitates the transport of a number of ions and other material again is concentration gradients into the vacuole in plant cells. Tonoplast. Concentration of these substances significantly higher in the vacuole in comparison with cytoplasm, ions and other materials. Cytoorganelle, which is considered as a powerhouse of cell. Cytoorganelle, which is considered as a powerhouse of cell, mitochondrion. Why the concentration of ions and other materials are more in vacuoles than in cytoplasm? Tonoplast facilitates the transport of a number of ions, and other materials against concentration gradients into the vacuole. Concentration gradients into the vacuole. Function of contractile vacuole. Osmoregulation and excretion. Contractile vacuole, example, amoeba, euglena, paramecium. Structures formed after engulfing the food particle. Food vacuoles. Food vacuole in the cells. Example, protist. Cytoorganelle, which is not easily visible under the microscope unless specifically stained, that is mitochondria. Majority of the chloroplast of the green plants are found in the dash cells of the leaf. Mesophyll. The number of mitochondria per cell is variable depending on the dash of the cell, physiological activity, and shape, size. Physiological activity, shape, and size. Alloplast store oils. They are composed of ribonucleic acid and proteins and are not surrounded by any membrane, ribosomes. Typically, it is sausage shaped or cylindrical, having a diameter of 0.2 to 1.0 millimicron, average 0.5 millimicron, and the length 1.0 to 4.1 millimicron, that is mitochondria. The granular structures which first observed under the electron microscope as dense particles by George Paladin are ribosomes. Double membrane bound structure with the outer membrane and inner membrane dividing its lumen distinctly into two aqueous compartments, that is mitochondria. Double membrane bound cytoorganelle and of the two and inner membrane is relatively less permeable. Chloroplast. Structures of potato which store carbohydrate. Amyloplast. Amyloplast. The two aqueous compartments of mitochondria are outer compartment and inner compartment. Outer compartment and inner compartment. Amyloplast store carbohydrates. The compartment of mitochondrion, which is filled with a dense homogeneous substance called the matrix. Inner compartment. Alloplast store proteins. Membrane of the mitochondrion, which form the continuous limiting boundary of the cytoorganelle. Outer membrane. This gives the part of the plant yellow, orange or red color. Chromoplast of plastids. Chromoplast of plastids. Membrane of mitochondrion, which form a number of infoldings towards the matrix. Inner membrane. Colorless plastids, which have varied shapes and size with stored nutrients. Leucoplast. Infoldings of the inner membrane of mitochondrion are called cristae. Number of mesophyll cells in chlamydomonas, 1. Infoldings of the inner membrane of mitochondrion which increase the surface area are called cristae. In the dash, fat-soluble carotenoid pigments like keratin, xanthophyll and others are present in chromoplast. Location of enzymes which are associated with the mitochondrial function. Location of enzymes which are associated with the mitochondrial function. Two membranes of mitochondria. The chloroplast contain dash. The chloroplast contain chlorophyll and carotenoid pigments. Chlorophyll and carotenoid pigment. Sites of aerobic respiration are mitochondria. Based on the type of pigment, plastids can be classified into chloroplast, chromoplast, and leucoplast. Type of DNA present in the matrix of mitochondria. Single circular DNA. Cytoorganelle which bear some specific pigments, thus imparting specific colors to the plant. Plastids. Type of genetic material in the mitochondria. Single circular DNA and single circular DNA and few RNA molecules. These are easily observed under the microscope as they are large. Plastids. Type of ribosomes in the mitochondria. 70S ribosomes are present. Cytoorganelle, which is present in all plant cells and in euglenoids, that is plastid. Type of division in mitochondria, fission. Lens-shaped, oval, spherical, discoid or even ribbon-like organelles having variable length 5 to 10 millimicron and width 2 to 4 millimicron, that is chloroplast. Number of chloroplasts in the mesophyll per cell in the green algae 20 to 40. The space limited by the inner membrane of the chloroplast are the space limited by the inner membrane of the chloroplast is called stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in stacks like the piles of coins called grana. Flat membranous tubules which are connecting the thylakoids of the different grana are called stroma lamellae. Grana is also called integral intergranal thylakoids. The membrane of the thylakoids enclosed in a space called lumen. Enzymes required for the synthesis of carbohydrates and proteins are present in stroma of chloroplast. Type of genetic material in chloroplast. Double-stranded circular DNA. Double-stranded circular DNA. Type of ribosomes in the chloroplast. 70S ribosomes. Chlorophyll pigments are present in thylakoids. The ribosomes of the chloroplast are smaller, 70S, than the ribosomes of the chloroplast are smaller than the 80S ribosomes. Type of ribosomes in eukaryotes. 80S ribosome. Type of ribosomes in prokaryotes. 70S ribosome. Number of subunits in each ribosome. 2. Subunits of 70S ribosome. 50S and 30S. An elaborative network of filamentous proteinaceous structure consisting of microtubule, microfilaments, and intermediate filament present in the cytoplasm is collectively referred to as cytoskeleton. Subunit of 80S ribosome, 60S and 40S. Subunits of ribosome and ribosomes are measured in Swedberg's unit. Sedimentation coefficient is Swedberg unit. Sedimentation coefficient is Swedberg's unit. Is called sedimentation coefficient. The dash in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell, that is cytoskeleton. It is indirectly a measure of density and size. Sedimentation coefficient. Hair-like outgrowths of the cell membrane are called cilia and flagella. Small structures which work like oars, causing the movement of either the cell or the surrounding fluid are cilia. Comparatively longer and responsible for the cell movement. Flagella. Flagella of prokaryotic bacteria, flagella of eukaryotic bacteria. Structurally same or different? Different. Electron microscopic study of a cilium or the flagellum show that they are covered with plasma membrane. Core of flagellum is called axoneme. Nucleus of a cell organelle was first described by Robert Brown. Part of flagellum which bear a number of microtubules running parallel to the long axis is called axoneme. Material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. Number of peripheral microtubular doublets which are arranged radially in axoneme 9. Number of centrally located microtubules in the axoneme 1 pair. Nucleus of a cell, when it is not dividing, it is called interphase nucleus. Arrangement of axonemal microtubules is referred to as 9+2 array. Highly extended and elaborated nucleoprotein fibers called chromatin. Number of nucleoli in nucleus, one or more. Central tubules are connected by bridges and is also enclosed by a central sheath which is connected to one of the tubules of each peripheral doublets by a radial spoke. Electron microscopy has revealed that nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nm, is called the perinuclear space. Interconnections of the peripheral doublets is possible with the help of linkers. Barrier between the materials present inside the nucleus and that of the cytoplasm is nuclear envelope. Nuclear envelope, we call nuclear envelope with perinuclear space. Both the cilium and flagellum emerge from centriole-like structure called basal bodies. Outer membrane of the nucleus. Outer membrane of nucleus usually remains continuous with dash endoplasmic reticulum. Outer membrane of the nucleus remains continuous with the endoplasmic reticulum and also bear endoplasmic reticulum which also bear ribosomes on it. At a number of the places the nuclear envelope is interrupted by minute pores which are formed by fission of its two membranes. The passages which makes movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm are called nuclear pores. Organelle usually containing two cylindrical structures called. Organelles usually contain two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials, centrioles. How the centrioles in a centrosome arranged each other? Perpendicular each other. Organization of the centriole in centrosome look like cartwheel. Highly extended elaborate nucleoprotein fibers are called chromatin. Highly extended elaborate nucleoprotein fibers are called chromatin. They are made of 9 evenly spaced perinuclea. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. They are centrioles. Each of the peripheral fibrillin centriole is triplet. The nuclear matrix of the nucleoplasm contain nucleolus and chromatin. Adjacent triplets of centrioles are linked. Central part of the proximal region of the centriole is also proteinaceous and called hub. Site for active ribosomal RNA synthesis is nucleolus. Hub is connected with the tubules peripheral triplets by radial spokes made of protein. Spherical bodies present in the nuclear matrix of nucleus are called nucleoli. The centriole form the basal body of cilia and flagella. Matrix of nucleus is called nucleoplasm or nuclear matrix. Spindle fibers that give rise to spindle apparatus during cell division in animal cell is formed by centrioles. Normally, there is only one nucleus per cell. Variations in the number of the nuclei are also frequently observed. Yes, true. The mature cell even lack nucleus in many mammals is RBC, erythrocyte. A mature plant cell which lack nucleus, sieve tubular cells of vascular plants. Spherical structures present in the nucleoplasm, nucleoli. The content of nucleolus continues with the rest of the nucleoplasm and it is not a membrane bound structure. It's not a membrane bound structure. Larger and more numerous nucleoli are present in cell for actively carrying out protein synthesis. Loose and indistinct network of nucleoprotein fibers called chromatin cell show structured chromosomes in place of the nucleus during different stages of cell division. Nuclear material in the chromatin is DNA. Basic proteins of chromatin are histones. Chromatin visible in the dividing cells as chromosomes. Histone and non-histone proteins, DNA and little RNA is the composition of chromatin. Length of long thread of DNA in human cell 2 meters. 2.2. Every chromosome essentially has a primary constriction at the centromeres and the sides of the disc-shaped structures are called kinetochores. Centromere holds 2 dash of a chromosome, chromatids of a chromosome. Chromosomes can be classified into 4 types based on the position of centromeres. If centromere is in the middle and form 2 equal arms in the chromosome, such chromosome is called metacentric chromosome. If centromere is slightly away from the middle of the chromosome, resulting in one short arm and one long arm, such chromosome, submetacentric chromosome. Centromere situated close to its end, forming one extremely short and one very long arm, acrocentric chromosome. If chromosome has a terminal centromere, such a chromosome is called telocentric chromosome. Sometimes a few chromosomes have non-striating secondary constrictions at a constant location. This gives the appearance of a small fragment called satellite. Many membrane-bound minute vesicles that contain various enzymes which are present in both plant and animal cells, microbodies. So that's it, the cell, the unit of life, and revise it properly. And second time when you are revising, put in double speed to answer it fastly before they appear on the screen. And then thank you.